us to the last phase, and that's the coagulation phase. The coagulation phase is like typically what people think of as blood clotting. Like when people say blood clotting, they're really talking about what's happening in the coagulation phase. Okay, so during the coagulation phase in general, what's happening is that the blood is changing from a liquid, right, a nice thin free-flowing liquid, to a thick gel. And we want it to form this thick gel so that we stop losing blood, right? We want to form this nice clot, this nice solid patch, so that we stop losing blood through that tear in the vessel. The way that we do that is through a bunch of chemical reactions, but at the end, the key point to all of the chemical reactions is that we convert something called fibrinogen into fibrin. Again, there's tons of clotting factors, a bunch of chemical reactions, but the whole point is this conversion of fibrinogen into fibrin. So the reason that that's important, and we'll get to this in a second, oh, here we go, right here. Convert circulating fibrinogen into fibrin. The reason that that's important is that fibrinogen is soluble in the blood. Free flows through the blood, soluble, no worries. Fibrin is insoluble. So once we convert the chemical fibrinogen into fibrin, now we have these strands that are insoluble. They're not soluble in the bloodstream anymore. And what happens is these fibrin strands will form kind of a meshwork. Think of it kind of like a screen, like on your window, like a screen. And then what happens is the red blood cells and platelets will all start to get trapped, mostly red blood cells, but they'll get trapped in that fibrin meshwork. And that forms a nice solid clot, a lot more stable than that platelet plug was. All of these chemical reactions that occur during coagulation, we call cascade reactions. And if you think about a cascade like a waterfall, right, you'll have like a little pool of water and then it crashes down, and then a little pool of water and it crashes down, and a little pool and it crashes down, right? And one thing kind of leads to the next. And that's how these reactions go. One reaction will make a product that stimulates the next reaction. That will make a product that stimulates the next reaction. So all of these chemical reactions just kind of lead to each other as you go down. And again, the whole point is that we want to end up making fibrin. When we look at these chain reactions, again, we're utilizing clotting factors. And we'll look at a list of them in a second. But these clotting factors include calcium. The calcium ions, just general calcium ions, are important. They stimulate two different pathways, the extrinsic and intrinsic pathway. Utilize calcium as a clotting factor. There are some enzymes involved, but more importantly, we utilize a lot of proenzymes. A proenzyme is inactive. It doesn't work, it doesn't do anything. So we have a lot of these clotting factors or proenzymes that are just floating around in our bloodstream, but they're not able to actually do anything yet. They can't catalyze any reactions. We have to activate them first. So we convert the proenzyme into an actual active enzyme, and then that active enzyme will go on to catalyze the next reaction. So a lot of our clotting factors we'll see are proenzymes as opposed to actual enzymes. So here you see a list of the clotting factors. I won't ask you to like reproduce a list of these or anything. As we go through the three pathways coming up, I'll bold the things that I'll ask you, that I expect you to commit to memory. You do see that calcium ions are on here though. Tissue factor we mentioned, right? Tissue factor coming from the vessels. What else? Platelet factor 3 we talked about before. Oh, hemophilia. So, in general, what happens with hemophilia?
We don't clot.
You bleed, right? You don't clot, you bleed. I'm sure you've all seen like some sort of a TV show where someone's a hemophiliac and you bleed longer. Your blood doesn't clot, so you bleed and bleed and bleed and bleed, and you can lose a lot of blood because of that. Hemophilia is a deficiency in factor VIII. Okay, so you do not make factor VIII appropriately, so you're not able to conclude the pathway that utilizes factor VIII, so it's really hard for your blood to clot. This is, in most cases, inherited. You can have a spontaneous mutation, but in most cases it's inherited, and it's what we call an X-linked recessive trait. So that tells us a few things. The DNA that codes for factor VIII is on the X chromosome, right? So women have what chromosomes? What two? Two X's, right? So women have X, X. What do you guys have? XY. Okay, so the DNA that codes for factor VIII that we need in clotting is linked to an X. Okay, so on here, on here, on here. That's where we have that DNA. It's recessive. What does that mean? Like dominant versus recessive. Think like recedes. It like recedes to the dominant. So if you have two of the same gene here, this one's recessive and this one's dominant, the recessive one gets covered up by the dominant one. So this is recessive. So that means that if you're a girl and say the X chromosome from your dad did not have the trait, it was perfectly fine. That was a healthy X chromosome. But the X chromosome from your mom did have it, and this is a problem chromosome, it doesn't matter because this is going to cover that one up. The bad one is going to recede. So you can carry the bad gene and not actually have the disorder because your good healthy X has a good copy for how to make factor VIII and you're fine. But if you're a guy, the guy only has one X. So there's no covering it up. This is the only place that you have the DNA that tells you how to make factor VIII. There's no backup plan. So guys aren't carriers. If a guy has a deficient or inappropriate gene for factor VIII, he has hemophilia, symptomatic hemophilia. So it's much more common in men, much more common in men than it is in women because they don't have that extra X chromosome to cover up that recessive trait. The treatment for it is we give just IV factor VIII. We make it. We used to give it naturally, like we used to transfuse people with human blood, but we make factor VIII in the lab now, which is a lot safer, and we just give it IV. Okay, sorry, I got way off base. What's up?
In the three phases, is there any overlap?
Mm-hmm. Yeah, we'll go through it now, but the extrinsic and intrinsic essentially are like progressing at kind of the same time. One's a lot quicker than the other, but they're both kind of happening and then they lead to the same spot.
I meant with vascular, platelet, and coagulation.
Oh yes, there's overlap. Yeah, they all kind of begin at the same time.
Oh, they do.
Yeah. Relative, I mean like within seconds.
Okay.
Quickly. Okay, so we'll kind of finish up by talking about the coagulation pathway in more detail. Right? We had the vascular phase happening, we had the platelet phase happening, we shrunk the vessel, we made the platelets really sticky to try and plug it up, and now we want the blood to actually clot. We want a nice, stable, long-term clot to form to plug it up while we repair the vessel. Okay, we do that via coagulation. Again, the whole point here is we've got a ton of chemical reactions and we want to convert fibrinogen into fibrin. Why? Fibrin is insoluble, it makes the mesh screen. Fibrinogen soluble. Okay, once we convert it to fibrin, it is insoluble in the blood. It forms that nice substructure for us. Again, coagulation itself, just this coagulation has three pathways. Okay? the extrinsic pathway, the intrinsic pathway, and then we'll see that both of these lead us to the common pathway. So we'll kind of have like extrinsic, intrinsic, common, like that. So the extrinsic pathway is called extrinsic because it begins outside of the blood. Okay, like the vascular phase was happening in the actual vessel. This begins outside of the blood in either the tissue or the vessel. What happens with the extrinsic pathway is that that tissue factor, which was factor 3, leaks into the bloodstream. It combines with multiple other factors, and together they catalyze the reaction to produce factor 10. Again, remember I said calcium was involved in both the extrinsic and intrinsic. Calcium is really important for clotting. So again, the extrinsic pathway is starting outside of the bloodstream. Remember when we break that vessel wall, we said the vessel wall was going to release tissue factor, or factor 3. That tissue factor, once it leaks into the bloodstream, combines with multiple other things. You don't have to memorize, but factor 7 calcium. And then that activates factor 10. Okay, so it utilizes tissue factor, and we end up activating factor 10. The intrinsic pathway, which is a bit slower of a pathway, but leads us to the same place. The intrinsic pathway occurs when those exposed collagen fibers, remember I said whenever we tear a vessel, some of the rebar structure sticks out, right? Some of those collagen fibers are exposed and they stick out. Well, that lets the body know there's a problem, right? The collagen fibers should not be exposed like that. And when they are, that starts to activate proenzymes. And this can go a couple of different ways. Usually it's 8, usually it's 12, but it can kind of go with multiple different factors here. It's not always the same factor that gets activated. One of the keys here, though, is that this is assisted by platelet factor 3. Where was that released? It's really easy. Platelets. So platelet factor 3, you're thinking of tissue factor. Platelet factor 3 is going to kind of catalyze this. So we're starting in the blood for intrinsic. We activate proenzymes to active enzymes. We utilize platelet factor 3. That gets added to the bunch. We've got this whole series of reactions. This is where 8 normally comes in. So this is where hemophiliacs would have a problem. But we have a whole series of reactions, and then again, it culminates with the activation of factor 10. Okay, so it culminates with the same exact thing that the extrinsic pathway culminated with, activating factor 10. After we activate factor 10, that's when we start the common pathway. So again, I was saying how they both lead to the common pathway. That's activating factor 10, and then the common pathway goes down from there. I'm going to erase this so you can actually see it. So the common pathway is where the other two pathways come together. Right? Both of those will activate 10, and activated factor 10 gives us prothrombinase. What does ACE mean? It's an enzyme, right? So prothrombinase is an enzyme that works on prothrombin. Pretty easy. We activate 10 into prothrombinase. Prothrombinase converts prothrombin into thrombin. That terminology makes sense, right? Pro, like before, like pre, or gives rise to. Prothrombin gives rise to thrombin. How does it do that? How do we activate the inactive prothrombin into an active thrombin? With prothrombinase. Okay, so prothrombinase catalyzes the inactive prothrombin. Let's write that here. Inactive Right? That's just how it floats around in your bloodstream. You don't want clots like crazy, so it floats around not working. It's inactive. Okay? But we activate it, and then it activates into thrombin, which is an active enzyme. Okay, so now this thrombin can go catalyze another reaction. It's the thrombin that catalyzes fibrinogen into fibrin. Hey, and what did we say about those? Fibrinogen is what? Fibrinogen is what? And fibrin is insoluble. I started writing. And then again, we said that fibrin is this meshwork, right, that traps the other blood components to form a nice clot to stop the bleeding. This seems like much more than it is.
Why isn't it called thrombin? It's an enzyme.
Well, I think because it's working on prothrombin.
Okay.
It's not working on thrombin. I mean, I think they would have been.
Well, I mean, it converts to the enzyme thrombin. So
Yeah.
still a question, but
Okay. Okay, so this is the coagulation phase. Okay, again, we're trying to talk about how we stop bleeding. We do that via the vascular phase, right? The platelet phase, and then the coagulation phase. In the vascular phase, we utilize endothelins, right? Those endothelins tell the vessel to constrict to get smaller. The endothelial cells get really sticky. They contract to expose the basement membrane. So now we have a smaller little hole. We tell the endothelial cells to divide. We're trying to kind of just make the hole as small as possible. During the platelet phase, we activate the platelets, right? And we do, they attach and then they aggregate. So adhesion and aggregation. The platelets will attach to the exposed collagen, the basement membrane, the endothelial cells, and then they all start to stick to each other. That forms a platelet plug to try and temporarily patch up that hole. Platelet plugs are not stable, so we need a more long-term solution. That's where the coagulation phase comes in. Okay, it's really quick to make the platelet plug, but it doesn't last long. This takes longer, coagulation takes longer, but it lasts longer. So during the coagulation phase, we have the extrinsic pathway, the intrinsic pathway, and the common pathway. Okay, we start with the extrinsic and intrinsic pathway. They both kind of start at the same time, and they're both going to lead to the common pathway. Extrinsic, remember X, think like exit, right, or external, that's somewhere outside. So the extrinsic pathway starts outside of the blood, typically in the broken vessel, okay, the damaged tissue. Remember we said that this pathway is going to be kind of stimulated by tissue factor, which is factor 3, which makes sense, right? We're talking about damaged tissue, tissue factor. We also utilize calcium, which we're going to use in the intrinsic pathway as well. We have other clotting factors that come into play, and eventually we activate factor 10. The intrinsic pathway is in because it begins in the blood with the platelets. Okay. Again, we utilize platelet factor 3, and we also utilize calcium. We've got multiple other clotting factors that come into play. And eventually we do the same thing. We activate factor 10. And that's where the common pathway starts. An active factor 10 gives us the enzyme prothrombinase. Prothrombinase works on prothrombin. Converts prothrombin to thrombin. Thrombin's an active enzyme. Thrombin converts fibrinogen into fibrin. Fibrin is insoluble, so it It's not soluble in the blood, does not go into solution. So it forms that nice meshwork, or red blood cells start to get trapped in it. That forms a nice, stable, long-lasting clot. This pathway is super, like these terms are really, really important. Okay, the rest of it I kind of try and simplify as much as possible, but those are important. Yes?
Extrinsic and intrinsic pathways happen simultaneously.
Not at the same speed, but yes, simultaneously. This takes longer, but they do both start at the same time. It's just there's different reactions going on, so they take different lengths of time to finish.
Is that one way that plasma is different than interstitial fluid? Is that it has fibrinogen?
That's one of the plasma proteins.
Okay.
So when we say that plasma has plasma proteins that interstitial fluid does not have, one of those plasma proteins that it has is the fibrinogen. So yeah, you do not see that fibrinogen in the interstitial fluid. Yep, that's good. And here you can kind of see that, right? You can see this meshwork that we make, a little bit messier than my perfect grid, and then the red blood cells getting trapped in it. And I'm sure you guys have seen that happen in your own blood before. So thrombin is the second area where we see positive feedback happening. Remember we said that the platelets were involved in their own little positive feedback, where activated platelets got sticky, released chemicals, activated more platelets. They got sticky, released chemicals, activated more platelets, and it was this little positive feedback loop. The platelet factor also initiates this feedback loop in relation to thrombin. So what we see happening is that thrombin, remember, stimulates the release of tissue factor, which is 3, and platelet factor 3. Both of those lead to the production of more thrombin, right? Remember, tissue factor 3 goes through the extrinsic pathway, and that ends up leading to the common pathway, which activates and produces thrombin, right? That was tissue factor 3. Platelet factor 3 stimulates the intrinsic pathway. That ultimately leads to the common pathway, which activates thrombin. Thrombin causes the release of more tissue factor and more platelet factor. Tissue factor stimulates more thrombin. Platelet factor stimulates more thrombin, right? Thrombin stimulates the release of both of them. So it's this positive feedback loop that results in more and more and more thrombin being created, which causes more and more and more fibrin, more and more clotting. Interestingly enough, thrombin will also stimulate the breakdown of clots, which we'll see in 1 second. You guys can cross this off. I had this as like an FYI. Normally I talk through it, but we just don't have enough time for me to go off on my tangent. Okay, so fibrinolysis. What do we form a clot out of? What's the protein called that makes the clot? The insoluble one. Fibrin, right? What does it mean to lyse something? To break it apart. Fibrinolysis is lysing the fibrin. It's literally busting the clot apart. Okay, because we made the clot for a reason. But eventually the vessel repairs itself, and we don't want that clot there any longer. Once it gets old, it'll start to detach and break apart, and if it goes free through the vessels, it can get clogged places and cause problems, and it blocks vessels. So after we don't need that clot anymore, we want to digest it. We want to break it apart. That's the process of fibrinolysis. Okay, this is a relatively slow process. The chemical reactions that are involved take longer. The breaking down of the fibrin takes longer. That's on purpose. That gives us time for the vessel to actually heal before the clot gets broken up. We see that fibrinolysis is stimulated by thrombin and TPA. TPA is tissue plasminogen activator. If you work in an ER, you've probably heard the term TPA before. But both of these are going to go on to activate a proenzyme Are proenzymes active or inactive? Inactive, called plasminogen. Okay, so this inactive plasminogen is going to get converted into plasmin. Plasmin is active. And it's this plasmin that actually breaks up the fibrin clot. So thrombin can activate plasminogen into plasmin and break up a clot. TPA can activate plasminogen into plasmin and then break up a clot. It's weird because thrombin formed the clot, right? And doesn't that seem weird? I've got this enzyme that's doing both, building a clot and breaking up a clot. It seems like it would just cancel each other out, right? But the thing is, the chemical reactions happen at much different speeds. The reactions to build the clot, to convert the fibrinogen into fibrin and form the clot, are fast. This is slow. The actual process of converting plasminogen into plasmin is a slower reaction. The actual breaking down of fibrin by plasmin is slow. It takes a long time. So thrombin does both, but it forms it super fast, and then we slowly, super slowly break it down. Okay? So don't be confused by that. They do not cancel each other out. The speeds are much different. TPA is cool, tissue plasminogen activator, or TPA. We use that pharmaceutically. That's what we use like during a thrombotic stroke. When someone's having a stroke and we want to actually bust up the clot, we do that with TPA. Do you know, if you didn't have me last year, do you know what a thrombotic stroke is? No? Okay. So this is your brain. Your brain has all these vessels that deliver blood to it, right? The blood's really important. It has to get to all areas of your brain, or your neural tissue dies pretty rapid. So sometimes we have clots that form that should not be there. Like for example, if we don't dissolve one fast enough and it breaks off, or if you just form more clots than you should. You can have clots floating around your bloodstream when they shouldn't be there. When they get to the tiny little vessels in your brain, they can clog them. Okay? If we have a clot that's lodged right here in this vessel, now none of this area over here is getting blood anymore. And that tissue dies. That's a stroke. This is a thrombotic stroke because this is a thrombus. The clot is a thrombus. So it's a stroke that's caused by a clot, by a thrombus. So one of the things that we can do to try and fix this is to break up that clot, right? If I do it fast enough, then I restore blood flow, right? And this area of the brain is now happy again, and you can kind of recover some of that lost function. You gotta do it quick though. So what we do when somebody has a thrombotic stroke is we give TPA. We can actually give IV TPA. That goes in and busts up the fibrin, and that can restore blood flow. It's super important that you make sure it's a clot that's the problem. Some people have a stroke because of a bleed, and then you would just make it worse if you gave them TPA. And TPA's got to be given super fast. If the stroke, that's why it's like they say rapid response for a stroke. Time is brain. It's got to be given super quick. They say that after a few hours, the risks are greater than the benefits. But that's what TPA is. It's busting up the fibrin clot. So last thing, your diet can affect your ability to form clots. We need calcium ions, remember, in both the extrinsic and intrinsic pathway. They act as clotting factors, so you have to have enough calcium. You also need vitamin K. Vitamin K is necessary for us to make some of our clotting factors. So if you don't have vitamin K, you're unable to make some of your clotting factors. Vitamin K is produced in the intestine, but we also ingest it. It's mostly in green leafy veggies. It's in a lot of different stuff, but green leafy veggies are high in it. So spinach, kale have high levels of vitamin K. The reason that this is important is, again, pharmaceutically. Have you heard of a drug called Coumadin or warfarin? That's a vitamin K antagonist. Blocks vitamin K. Without vitamin K, you can't make clotting factors. So it's an anticoagulant. It decreases the clotting factors that you have, thus decreases the clots that you throw. And vitamin K is important because you can actually adjust how effective your warfarin is by adjusting how much vitamin K you eat. So if you're on like 2 milligrams a day of warfarin and it works for you, and then all of a sudden you start eating a spinach salad every night, now you have much more vitamin K and you need to block all of that vitamin K. So you're gonna need much more warfarin in order to do it. Okay, so vitamin K intake's really important when you'll have like, patients that are on warfarin. Heparin is another anticoagulant. It does not work at vitamin K though. It activates something called antithrombin, and that heparin activates thrombin. Completely different. Okay, I'm only an hour behind.